my name is Alex the Polymath, and this is my presentation on DNA. And its title is DNA: Description and Discovery. So who discovered DNA? Well, the scientist pair of James Watson and Francis Crick are the scientists that are credited as the discoverers of DNA, but it's kind of a controversy whether they are or not. Actually, they weren't the ones that discovered it at all. There's not a controversy about that. It was actually another scientist named Frederick Miescher that discovered it. Okay, but anyway, they weren't the ones that discovered DNA itself. They just discovered that DNA was made of a double helix. Yeah, so here are James Watson and Francis Crick. So, continuing on, who discovered DNA? Well, a scientist named Frederick Miescher, as I mentioned before, was the person who discovered DNA. Yeah, he discovered it way before James Watson and Francis Crick. And he did not call it DNA though. He called it a nucleon. And then he also called it a nucleic acid after that. The point is that he called it a nucleon rather than deoxyribonucleic acid, or DNA as we know it today. And then there were a bunch of other scientists that built upon his research. Then some of these include Watson and Crick and Rosalind Franklin and Maurice Wilkins. There's a bit of a controversy on whether Watson and Crick borrowed information from Rosalind Franklin and Maurice Wilkins without asking. It's a bit controversial. There's actually this TED Ed about Rosalind Franklin. There's this cool TED Ed. You can watch that if you want. So this is the guy I just mentioned, Frederick Miescher, the guy who discovered DNA. And this is Rosalind Franklin. The sad thing is about Rosalind Franklin is that she died at such an early age. So she didn't get a Nobel Prize like Watson and Crick. So it's hard to know whether she would get a Nobel Prize if she had lived. Well, didn't die at that young age of 30. Okay, anyways, let's go on to this description part of the presentation. So what is DNA made of? Well, DNA is made up of 4 different chemical bases. They are adenine, which is abbreviations A, and it has this chemical structure here: 3 nitrogens, a nitrogen-hydrogen, and a nitrogen with 2 hydrogens. Thymine, with abbreviated T, with 2 nitrogen hydrogens and 2 oxygens. Cytosine, with nitrogen, oxygen, a nitrogen hydrogen, and a nitrogen double hydrogen. And finally, guanine, abbreviated G, with 2 nitrogens, 2 nitrogen hydrogens, 1 oxygen, and 1 nitrogen double hydrogen. Sorry about that. So, how much DNA is there in the human body? This might be a question that you're wondering. In the human body, there are 3.2 billion bases of DNA. I know, crazy, right? You wouldn't think in human body there'd be that much, but because DNA is so small that you'd be able to fit it all in your human body. So, how do chemical bases bond, though? The chemical bases, by the way, I forgot to mention this. The chemical bases would be like adenine, guanine, thymine, and cytosine. How did they bond? Well, it's actually pretty simple how they bond. Adenine bonds with thymine, and cytosine bonds with guanine. And this is all the time. This happens at all times. There's no exceptions. And how are these bonds staying connected, though? Well, These bonds are connected by hydrogen. Yeah, the bonds are made of hydrogen. Yeah, and then you can, on the right, you can see basically a diagram on how all of this works. So what is the difference between DNA and RNA? Well, DNA has a double helix while RNA has a single helix. DNA has thymine in its 4 chemical bases, while RNA has uracil instead. And DNA has this molecule with all of those right there. I'm not going to read it because it's not necessary for me to read it. But yeah, the codes are slightly different. And DNA is a pair of nucleobases, while RNA is a single nucleobase. And here is an image describing it all. So here are my sources that I use. Thanks for listening.